Okay now, students, as we keep moving through the arterioles, I'm going to tell you a little bit about our next stop, the capillaries. This is where the real fun begins. The capillaries are vessels that supply the tissue with us.
So, just how big are the capillaries?
Perfect question. Now that we are reaching the terminal arteriole and about to enter the capillary bed, capillaries are the smallest blood vessels in the body. They are so small that blood cells like us can typically only pass in a single file line. Which means, come on everybody, single file. Here we are, the capillary bed. The capillaries are the exchange vessels connecting the arterioles and the venules. The arterioles, as I said earlier in our tour, are small arteries that deliver oxygen and nutrient-rich blood, like us, to the capillaries. Venules are small vessels connected to the capillaries that drain us into the veins to eventually be returned to the heart.
So, what makes the structure of capillaries so different than arteries and veins?
Fabulous question, student. Capillaries are the only vessels thin enough to let us pass through. This is because capillaries, unlike the arterioles we were just in, or the veins we will enter, do not have a tunica externa or media. They only have a basement membrane, and endothelium, also known as the tunica intima.
But I was told that the capillary bed consists of two types of vessels. Is that true?
Yes, it is. Capillaries consist of a vascular shunt, also known as a meta-arterial thoroughfare channel, and true capillaries. A vascular shunt is a short vessel connecting the terminating arteriole and postcapillary venule. It is interesting because it has the structural characteristics of both a capillary and an arteriole. It is slightly larger than a typical capillary. It forms smooth muscle valves, also known as sphincters, that monitor our permission into the true capillaries, the actual exchange vessels. If the sphincters are closed, we would go straight from the meta-arteriole to the thoroughfare channel into the postcapillary venule and eventually into the veins. Consider them your hallway monitors.
Wait, so where are we now?
We are in the vascular shunt. We can go through either the capillary into the tissues or through the channel into the postcapillary venule. We have the option to explore the true capillaries. What do you say, guys?
Yay! Let's explore the true capillaries.
Okay, great. Before we begin, I would like to describe the three types of true capillaries. They are the continuous capillaries, fenestrated capillaries, and sinusoid capillaries.
I know what the continuous capillaries are. They are the capillaries that have tight junctions and penocytic vessels that carry fluid across the endothelial cell. They only allow small molecules such as water and ions to pass through. They constitute the brain-blood barrier.
That is correct. Anyone know what fenestrated capillaries are?
Oh, me! Fenestrated capillaries have pores or fenestrations in the epithelial cells. They allow small molecules as well as some proteins to pass through.
Great job! Fenestrated capillaries occur in areas where absorption and filtration are important, such as the kidneys and small intestines. Sinusoid capillaries have large intercellular clefts and fenestrations with very few tight junctions. They are more irregularly shaped and have larger lumens than other capillaries. Due to their structure, they allow larger molecules to pass through their walls and enter the tissue. We move slowly here and in the capillaries to ensure that adequate exchange is occurring.
Are capillaries found in the same amounts in all tissue?
Most tissues have a rich capillary supply, except tendons and ligaments, because they are poorly vascularized. Cartilage and epithelial lack capillaries completely. Anyone else have any questions before we enter the true capillaries?
Yeah. How much of us can flow into the capillary at one time?
Well, that depends on the arterioles and how much they dilate or constrict. When arterioles dilate, we can move into the capillaries faster. When they constrict, it takes us longer to get to the capillaries. Intrinsic and extrinsic controls determine this. The blood flow to the capillaries is less when we were all in class than when we are all together, rushing to get places. Moving on. Can anyone tell me about the velocity of blood flow?
I think that there is a direct relationship between the velocity of blood flow and the total cross-sectional area of the vessels that we are moving through.
Actually, it is quite the opposite. Let's take a second to really think about this.
Well, it makes sense because as the total cross-sectional area increases, our flow would decrease because we would have a larger pathway to move through. That does make sense. I wouldn't have thought of it that way before.
Exactly. The total cross-sectional area is extremely important when discussing the topic of blood flow.
I have a question. How do molecules actually move through the capillaries?
There are different ways that molecules can move through the capillaries. They include lipid-soluble molecules like respiratory gases can diffuse through the lipid bilayer of the endothelial cell plasma membrane. Small water-soluble solutes can pass through fluid-filled intercellular clefts or fenestrations, and some larger molecules can actively be transported in pinocytic vesicles or caveolae.
You mentioned that there are these molecules that could be either diffused or actively transported. What do you mean by that?
Well, diffusion is the movement of materials across a semi-permeable membrane, and the molecules move from an area of high concentration to an area of low concentration. Diffusion is a form of passive transport because no energy is being used in order to move the molecules across the membrane.
Well, that makes a lot of sense. So active transport would be when energy is needed to move the materials. With that said, the molecules must be moving against the concentration gradient.
That's exactly right.
Are there other factors involved in our flow through the capillaries?
Why, yes, there are. Our viscosity and the area of the vessels themselves all play a role. Okay, class, it's time for recess. And when we come back, we will start discussing more about how these particular substances actually move across the membranes of the capillaries and about bulk flow and pressure systems.